Good morning and good evening, everyone. Can you hear me?
Yes, we can hear you.
Okay. So, myself, Boda Praveen from Department of Plant Pathology, University of Agricultural Sciences, Bangalore. So, my presentation is here, millets. So, small millets are one of the oldest food known to the humans and possibly the first cereal grains to be used. And they are the rich source of nutrients like calcium, magnesium, zinc. And this can be cultivated in any dry land or any situation. So, these are the hardy crops. So, we have to take care of these things. So, here, these millets offer threefold of security to the nation, like food security, nutritional security, as well as the economic security. Now, when it comes to the little millet is also called, it is locally known as Kutki and Same in Karnataka and the Indian states. But this crop is cultivated by tribal and poor farmers in the low fertile soils. It has excellent rejuvenating capacity and it is a rich source of some of the minor nutrients. But here the thing is, this nowadays this crop is affected severely with that leaf blight pathogen Alternaria alternata. So for this crop and this disease, there is no information available for this management and the characterization of this pathogen. These are the things. So here I taken as a challenge to work on this identification and characterization of this leaf blight of little millet, which is caused by the Alternaria alternata. And the outcome of my research, which we expected, is identification and characterization of this pathogen, enumeration and characterization of these toxins, which is produced by the pathogen, and the host and studies of the pathogen. Previously, there is no report on these millets, but now it started infecting some of the millets. So how far it is going to affect any of the millets? So for that, we have set an objective as a host and studies and identification of resistant source, and of course, the disease management. Now here, for identification of the pathogens, we have to take into considerations like morphological characters, some of the cultural characters, as well as the molecular characters. Based on the symptoms in the field, and as well as the cultural characters of the pathogen, as well as some molecular characterizations, we have discussed and we came to know that this is caused by Alternaria alternata. So the molecular characterization is done by the ITS. So this Alternaria has more than 300 species till now. So we cannot differentiate at the species level only with the ITS primer. So we have to go further with some of the genes like Alternaria alternata specific genes, histone proteins, and glyceraldehyde phosphate, LSU, SF. So these are some of the genes which we can differentiate from different species. So here, for this pathogen, as it is the new report on this little millet, we have gone through the whole genome sequencing by using the Oxford nanopore minion sequence. So here we have prepared the library in our Department of Plant Pathology. Later we sent it for the sequencing by EP2 mine that analysis, bioinformatic analysis. So this is the data we have achieved after this nanopore minion sequence, and this is a total basis which we got by this nanopore sequencing based calling. So here is the rough data which we have we have taken from that. So later the further analysis has been it is like under process. So here Alternaria, it is going to produce some of the deadly toxins which will help in the pathogenicity and causing the disease as well as the virulence. These toxins, it will start from the infection side to at the final process, increasing the disease severity. Many genes will contribute to the production of these toxins, the deadly toxins. From the infection side to after producing the different symptoms, the more and more toxins, around 70 different types of toxins are going to cause the disease, which provoke the disease incidence as well as the severity. So here, in order to know which genes which are which toxins majorly present in that Alternaria, we gone through the HPLC and we found that majority of these toxins are tenuagenic acid, Alternaria, or Alternarian monoethyl ether. These are the toxins which we have found in a major amount. So for host and studies, we have gone through different crops, and this pathogen is going to infect little millet, brown top, wheat, oats, and kodo millet. So we come to know the host strains of this pathogen is more. So we have screened for the resistant source, and we found around 30 resistant genotypes among 142, which we have collected from throughout the India, different states. So of course, we went for the in vitro studies by using some botanicals, bioagents, and fungicides. We got the eucalyptus, which inhibited the pathogen, as well as in case of bioagents, Bacillus subtilis, it infected, it reduced the pathogen, as well as some of the fungicides. They have reduced the pathogen. So thank you.